In this session, we will discuss about the definition and types of fermentation, which is an important topic from Unit 5 of Pharmaceutical Biotechnology of 6th semester B-Pharm. So before starting the session, I would like to explain some questions which is usually uh, seen from this area. There are some questions like write in detail about structure and parts of fermenter, its control with the neatly labeled diagram, and sometimes it is like define fermentation and explain the production of enzyme by bioprocess techniques. And as a 5 mark question, explain brief about the structure and part of fermenter with the help of diagram, and discuss design discuss the design features, merits and demerit of continuous fermenters, then operations of aerobic fermentation. Then as a 2 mark question, there are classify fermentation methods, different types of spargers, and sparger types and control of fermentation parameters. So these are the some of the topics which is usually seen from this area. So based on that, this is the learning objective of the session. After the session, the learner will able to understand, describe and write about the definition of fermentation, definition of bioprocess, definition of fermenter, then industrial products of fermentation and different types of fermentation. So this is the learning objective. Now let's start with the definition of fermentation. So what is mean by fermentation? Fermentation is a process or metabolic process by which the microorganism generate energy from organic compound by activity of some enzyme. So this is basically use of an microorganism to convert one compound to another compound by using some kind of enzyme activity which is produced by the microorganism. And the word fermentation originated from the Latin verb fervor, which means to boil. So basically in the olden era, they used to prepare something by boiling any natural products to get some related product from the natural originated products. Okay. So fermentation, it is mainly applied to the growth of microorganisms in liquid media under either aerobic condition or anaerobic condition to produce economically useful product. That is the basic thing of fermentation. We are growing the microorganism. So by growing the microorganism in an culture media, especially in the liquid media, the microorganism will produce some primary metabolites or secondary metabolites. Sometimes the primary metabolites are essential for the growth of the same microorganism, and secondary metabolites are not essential for the growth of microorganism. So these primary and secondary metabolic end products are usually useful for human being, so that we will use a microorganism to produce some kind of products by growing the microorganism in a culture media. That is the basic thing which will happen in the fermentation process. അപ്പോൾ fermentation എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ സിമ്പിൾ ആയിട്ട് പറഞ്ഞു കഴിഞ്ഞാൽ ഒരു കൾച്ചർ മീഡിയ വേണം, ഒരു മൈക്രോ ഓർഗാനിസം വേണം, ആ മൈക്രോ ഓർഗാനിസത്തിന് കൾച്ചർ മീഡിയയിൽ ഗ്രോ ചെയ്യാനുള്ള കണ്ടീഷൻസുകൾ വേണം. അപ്പോൾ pH അഡ്ജസ്റ്റ് ചെയ്യേണ്ടി വരും, ടെമ്പറേച്ചർ അഡ്ജസ്റ്റ് ചെയ്യേണ്ടി വരും, ഇങ്ങനെയുള്ള കാര്യങ്ങളൊക്കെ കൊടുത്തു കഴിഞ്ഞ് കഴിഞ്ഞാൽ മൈക്രോ ഓർഗാനിസം ഒരു കൾച്ചർ മീഡിയയിൽ ഗ്രോ ചെയ്യും. ഗ്രോ ചെയ്യുന്ന സമയത്ത് അത് അതിൻ്റെ ഒരു മെറ്റബോളിക് പ്രോഡക്ട്സുകൾ പ്രൊഡ്യൂസ് ചെയ്യും. സംടൈം പ്രൈമറി മെറ്റബോളിക് എൻഡ് പ്രോഡക്ട്സുകളായിരിക്കും, സംടൈം സെക്കൻഡറി മെറ്റബോളിക് എൻഡ് പ്രോഡക്റ്റ്. These primary and secondary metabolic end products are usually useful for humankind. So that is the basic fermentation. Now, so this is the metabolites. So during the fermentation process, the microorganism will grow in the culture media, and it will produce two type of metabolites, that is primary metabolites and secondary metabolites. So usually the primary metabolites will produce during the log or exponential phase. Okay, so we had discussed the growth curve of the bacteria. There is a lag phase, log phase, exponential phase and decline phase. So the primary metabolites are usually produced during the lag as well as exponential phase, and these metabolites are usually essential for the growth of microorganism as such. So these are the some of the example: glutamic acid, lysine, protein, vitamins, lipids, carbohydrates, citric acid. These are the some of the examples for metabolites, primary metabolites. Now coming to the secondary metabolites, these are produced during the deceleration or stationary phase, uh, which are not essential for the growth of microorganisms. Sometimes uh, at the end of the growth of microorganism will produce some kind of secondary metabolites like antimicrobial compounds, some enzyme inhibitors, some growth promoters. All these are not essential for the growth of microorganism, but they are essential for the human being. Okay, so these can be used for our economical purposes. Okay, now what is the difference between bioprocess and fermentation technology? So in olden eras we used to use the term fermentation for this process, but nowadays we replace the fermentation term with bioprocess technology, which is more recently used to replace the fermentation technology, because bioprocessing is broadly involved multitude of enzyme catalyzed reaction carried out by living cells or sometimes cell-free system. So in case of bioprocessing, we used to use some enzyme directly without using some kind of microorganism. Okay, that is the main difference. So while using fermentation also, a microorganism are producing some kind of enzymes, then that will produce uh, some reactions. So we can call it as a bioprocess as such. Fermentation is also a bioprocess, but bioprocess basically involve multitudes of enzyme catalyzed reactions. Okay, so the term bioprocess technology is to industrial use of higher plants and animals. That is another difference between fermentation as well as bioprocessing. In case of fermentation, we are using only microorganisms for this conversion of one compound to another compound. But in case of bioprocess technology, we will use some kind of higher plants or animal cells for the conversion of one compound to another compound. Okay, so now what is bioreactor or fermenter? So we had defined what is fermentation and we had defined what is bioprocess. Now coming to the fermenter or bioreactor. So these are the equipments which is used for either fermentation process or for bio processing. So bioreactors are basically devices in which we will use the organism are cultivated and motivated to grow to form and desired products. So its containment system designed to give right environment for the optimal growth and metabolic activity of the microorganism. And fermenter, which is usually refers to containment system for cultivation of prokaryotic cells only for bacteria and fungi. If we are using bacteria and fungi only for this process, that device is known as fermentation process. If we are using eukaryotic cells like mammalian cell or insects for this kind of bioprocessing, that equipment is known as bioreactor. So that is the difference. In case of fermentation, we will use only the microorganism like bacteria and fungi. And in case of bioreactor, we will use some eukaryotic cells like mammalian cell as well as insects. Now, what are the examples of industrial products of fermentation technology? So this is a kind of applications of fermentation process or fermenter. So first thing is we can use the fermentation technology for the production of foods like dairy products like cheese, yogurts, vitamins like B1, B12, etc. Then for the production of amino acid like glutamic acid, lysine, then production of glucose and high fructose syrup, then production of mushroom products, then bakery yeast, then food additives like antioxidant, colors and flavors, then beverages like beer, wine, whiskey, etc. So these are the some of the examples of products which can be used for the production of foods. And second examples, we can use fermentation process for the production of chemicals like organic compounds like ethanol, butanol, acetone, organic acid like citric acid, gluconic acid and lactic acid. And sometimes we can use the organic fine materials like enzyme, polymers like xanthan and dextrans. And sometimes we can use production of some inorganic chemicals like bioaccumulation of and leaching of copper as well as other metals. And also we can use fermentation technology for the production of some more pharmaceutical products or healthcare products like antibiotics, vaccines, steroids, diagnostic enzyme, monoclonal antibodies, and enzyme inhibitors. And also we can use it for production of some agricultural products like single cell protein, microbial pesticide, and compositing process plant cells and tissue cultures. So all these are some of the examples of products, industrial products of fermentation technology, and these are some of the common products and their microorganism and their applications. For example, ethanol. So this is a basic organic solvent which is mainly used for preservatives and production of many medicinal products, etc. So which is usually produced by fermentation process by using a microorganism Saccharomyces cerevisiae, so which is used as in beverages as well as industrial solvents, and glycerol, another sweetening agent which is used mainly in the syrup, etc. So this is produced by fermentation process by using Saccharomyces cerevisiae, and also this is used for nitroglycerin, which is an explosive. So this glycerol is treated with the nitrogen compounds to produce an nitrogen nitric acid as well as cellulose to produce nitrocellulose, which is an uh, explosive. So this can be used for production of explosive. And lactic acid, which is produced by Lactobacillus bulgaris, which is mainly used as in food and pharmaceutical products. Then acetone and butanol, which is produced by Clostridium acetobutylicum, which is used as a solvent. And it is also used for the production of some enzyme like alpha amylase by using Bacillus subtilis, which is mainly used for an enzyme which is used for hydrolysis of starch. So these are some of the applications or industrial products of fermentation technology. Now coming to the type of fermentation. So what are the different types of fermentation? We can classify the fermentation process based on their uh, addition of the nutrients, or sometimes based on their equipment nature, etc. So we will explain one by one. So first one is batch fermentation process. So as the name indicate, we will perform fermentation process as a batch, which means we will start an fermentation product and we will wait for finishing the fermentation process. Finally, we will get the product and then we will remove the products and then we will start the next batch. So like that, it is a closed culture system. So initial and limited amount of sterilized nutrient media is introduced into the fermenter at the initial stage, and then medium is inoculated with a suitable microorganism and incubated for a definite period of process under the optimum physiological condition. So by giving an optimum condition for the microorganism will undergo multiplication and passes through the different phases of growth phase and metabolism due to their change in the composition of the culture media and biomass as well as metabolites. So usually this process run for a definite period until a nutrient is exhausted. So here, which is mainly used, this biofermentation or batch fermentation process is mainly used for to produce the biomass as well as primary metabolites and secondary metabolites. So during the exponential phase, uh, there will be a production of primary metabolites. So in order to increase the yield of primary metabolite, what we have to do, we have to prolong the exponential phase. And uh, if we need any kind of secondary metabolites, so our desired product, if our desired product is an secondary metabolites, what we have to do, we have to reduce the exponential phase so that it will very fastly reaches to the uh, stationary as well as declining phase. So it will produce more yield of secondary metabolism. This is how we design the yield of fermentation products. So basically what we are going to produce, either the desired product is an primary metabolites or secondary metabolites. So if the desired product is an primary metabolites, we will prolong the exponential phase. So as the length of the exponential phase increases, there will be an increased production of primary metabolites. And If the desired product is a secondary metabolites, we will reduce the length of the exponential phase. So it will very fastly reaches to the stationary phase as well as decline phase, so that it will produce the maximum amount of secondary metabolites. So that is the batch fermentation process. We will it is a closed system. We will add the culture media at the initial stage only. In between, we will not add anything, and we will wait till the nutrient is exhausted. So that is the basic batch fermentation method. So that is the first method of batch fermentation method. Now let's see what are the advantages as well as disadvantages of this batch fermentation process. So basically, the advantage is we can reduce the contamination because we are going to inoculate only one required microorganism during the fermentation process. That also during the initial state of stages of fermentation process. Then we will close it. We will not open the fermenter till the nutrient is exhausted. Okay, and this is a very simple method of fermentation process, because we need a closed system, and we will add the nutrient as well as microorganism at initial stage, and we will wait till the reaction is over, and then we will open. So there is no complexity in this kind of fermentation process. But there are some disadvantages. Fermenter and equipments are to be cleaned and sterilized for every batch, so that will be very difficult. So after finishing production of one fermented products, we have to clean the entire fermenter, and we have to sterilize the equipments before starting the next batch. So this increased by eliminating turn around time as well as downtime. So it will require some time for cleaning, sterilization process, and usually the running cost is greater for preparing and maintaining the stock culture, because once we prepare the stock solution. What is mean by stock solution? This is nothing but the fresh inoculum which we have prepared. Okay, so in case of batch fermentation, if we need to produce the second batch, we have to wait till the first batch production is over. So again, we have to maintain the stock culture for the next production. Then increased frequency of sterilization cause greater stress and on the instrumentation as well as props. That is another disadvantage. Each time we have to sterilize, so that will cause some kind of damage to the entire equipments, and yield of the product will vary depending on the culture media we have taken, depending on the strain of microorganism is taken, and which require more personal requirement is there for the batch fermentation process. Okay, so these are the some of the advantages as well as disadvantages of the batch fermentation. Now coming to the second one, that is the fed batch fermentation. So as the name indicates, we will feed to the batch fermenter. What do you mean by feeding? So as we mentioned, in order to produce primary metabolites, we have to prolong the stationary log phase or exponential phase, right? So in order to prolong the log phase or exponential phase, we may have to add some nutrients in between the fermentation process before the fermentation process is over. Okay, so that is the feed batch fermentation. In this, substrate is added in increment at different time throughout the course of fermentation, and the growth of media is not removed until the end of fermentation. That is another point to be noted. We will only add the nutrients in between the fermentation process. We are not going to remove anything from the fermenter till the operation is over. Okay, so periodically substrate addition prolong the log phase as well as stationary phase, which may result in the increased cell mass. Consequently, produce the metabolites during stationary phase also increases. As it is difficult to directly measure the substrate concentration in the fed batch fermentation, other indicator that correlate the substrate consumption are used for measuring the amount of end product is formed. Okay, so basically the formation of organic acid, production of carbon dioxide, and changing pH may measured as an production of substrates, and accordingly we will add the nutrients. നമ്മൾ ബേസിക് ആയിട്ട് ഇതാണ് ചെയ്യുക. അവിടെ പ്രൊഡ്യൂസ് ചെയ്യുന്ന ഓർഗാനിക് ആസിഡ് മെഷർ ചെയ്യുക, അല്ലെങ്കിൽ ഗ്യാസ് കാർബൺ ഡൈ ഓക്സൈഡ് മെഷർ ചെയ്യുക, pH ചേഞ്ച് മെഷർ ചെയ്യുക, so that we can come to know whether the desired product is formed or not. Okay, so that is the basic fed batch fermentation. And this can be done in different types, like variable volume fed batch culture, which means same medium is added, resulting in increased volume. Another method is fixed volume fed batch culture. Very concentrated, very concentrated solution of limiting substrate is added at the very little amount. So that is a fixed volume fed batch fermentation. And cyclic fed batch fermentation, which is not usually possible to measure the concentration by following the direct method during the fermentation, and which is necessary for controlling the feeding process. Generally, direct method are employed, like variable fixed volume fed batch fermentation, or fixed volume fed batch culture method is used as an fed batch fermentation method. Now, what are the advantages as well as disadvantages of fed batch fermentation? So, based on the uh, fed batch fermentation, we can produce high density materials. We can control over the production by the product or catabolic uh, repression. We will discuss what is mean by catabolic repression later. And allow replacement of water loss by evaporation. Dealing with the toxic substance is very easy. Increasing antibiotic marked plasmid stability. No additional piece of equipment is required when compared with the batch fermentation. So only the difference is we will add some kind of nutrients in between the process. That is the only difference. And these are the some of the disadvantages. Not possible to measure the concentration of feeding substrate by following the direct method, and require precious analysis of microorganism to ensure exact growth of microorganism, require substantial amount of operator skill for setup of fermentation and development. Because based on the desired product, we may have to increase the concentration of the nutrient media, or sometimes we may have to decrease the concentration of the nutrient media. So basically the skill of the operator is very critical here to get the desired product. Now, third type of fermentation process is semi-continuous fermentation. So here the difference is a portion of nutrient media will be added and a portion of end product will be removed. So that is the difference when compared with the previous method. In case of batch fermentation, we will add the nutrient media initially and we will wait till the end of the fermentation process. And in case of fed batch fermentation, we will add some amount of nutrient media during the fermentation process, and we are not going to remove anything till the operation is over. But in case of semi-continuous fermentation, a portion of culture media is removed from the bioreactor and which is replaced with a fresh media. So the lag phase and other non-productive phases are very much shortened by using this kind of semi-continuous fermentation, and it is mainly used for the production of alcohol by fermentation process. And the advantages is the product output is much higher when compared with the batch culture, and there are some disadvantages like difficulty of handling bioreactor, long culture period which may lead to contamination, mutation and mechanical breakdown. So these are the some of the disadvantages of semi-continuous fermentation. Now last one is continuous fermentation. So this is an open system. During the process of fermentation process, we will continuously add the nutrient media and we will continuously remove the end products. Okay, as the nutrient media is added to the fermenter, same time from the opposite side we are going to remove the culture media continuously or remove the end product continuously with the replacement of some fresh sterile media. So both addition and removal are done at the same rate, so that the working volume remain constant during the entire process of fermentation. So this is a non-stop type of fermentation process. We are continuously producing the end product by giving nutrient media. Same time we are removing the used culture media or the end products. Okay. Now these are of two types: homogeneous mixed bioreactors and plug flow bioreactors. So the homogeneous mixed bioreactors are of two types: the chemostat bioreactors as well as turbidity bioreactors. So what is meant by chemostat bioreactor? The concentration of any one of the substrate is adjusted to control the cell growth and maintain steady state. That is the chemostat. So here the concentration of the chemical used is statics. We are going to adjust that to control the growth of microorganisms. And in case of turbidity bioreactor, instead of measuring the concentration of the nutrient media, we are just measuring the turbidity by using some kind of absorbance method to monitor the biomass concentration. So that is the turbidity bioreactor. And second type of continuous fermentation is plate flow bioreactor. Here culture solution flow through a tubular vessel without back mixing. So continuous ആയിട്ട് ഒരു ട്യൂബിലൂടെ പാസ് ചെയ്യുന്ന പോലെ we will add the culture media in one side and we will remove the formed end products from the other side. So that is a plate flow bioreactor. So these are the some of the advantages as well as disadvantages of continuous fermentation process. We can reduce the size of bioreactor very compact, because we don't need that much of bulk volume like 1000 liter or 5000 liter, because this will produce continuously the fermentation products. So we can add the nutrient media in one side and we can remove the product from other side. So the size of bioreactor will be relatively very smaller. Only little quantity of initial inoculum is added and no need of additional inoculation during the process. So in case of batch fermentation, after one batch we have to take fresh nutrient media, and to this we have to add fresh inoculum. But in case of continuous fermentation, an initial phase we will add some inoculum. That is enough for the entire process. We don't have to add much more microorganism. We just have to add the nutrient media, fresh nutrient media, as the used nutrient media is removed. Okay, then yield of the products is more consistent, because if we are adding and constant rate of or constant concentration of some culture media, it will produce the constant rate of an end product also. Okay, and downtime between the two successive fermentation for cleaning, preparing bioreactor is avoided in the continuous fermentation. No shutdown is required. So that is another advantage things. We don't have to give a break for the fermentation process, because we are doing this fermentation continuously without stopping. Once it is started, we will continuously add the nutrient media. And these are the some of the disadvantages. It is very difficult to maintain the sterility, because we are continuously running the fermentation, so that there is a chance of contamination by any kind of microorganism from the environment or from the person who are working there, or any kind of things or equipment used, or sometimes nutrient media used. Basically in fermentation we will use the natural products as an culture media, so there is a chance for contamination from the culture media also. So it will be very difficult to maintain the sterility, and the maintenance of same quantity of culture media for addition are difficult also, because we have to critically monitor the concentration of the end product during the fermentation process, in between the fermentation process, etc. And process become more complex and difficult to accomplish when compared with the batch fermentation process, because consistently we have to measure the different parameters which is used in the fermentation, with different concentration of nutrient media used in the fermentation. So these are the different types of fermentation process. Now so these are the classification based on the addition of the nutrient media. Now there are some another type of fermentation, like whether we are using air system or without air system. That is the aerobic fermentation or anaerobic fermentation process. So first one is anaerobic fermentation process. So as the name indicates, so this fermentation is carried out in the absence of oxygen. So there are two types of anaerobic microorganisms, obligative anaerobes as well as facultative anaerobes. So obligative anaerobes are the microorganisms which cannot withstand the oxygen or remain active only in the absence of oxygen, example like Clostridium species. And there are some facultative anaerobe which can able to withstand small amount of oxygen, but it will better grow in the absence of oxygen, like lactic acid bacteria. So these are some of the examples of anaerobic fermentation products, like Saccharomyces, like ethyl alcohol, Streptococcus, Lactobacillus, and lactic acid. So these are some of the types of different examples of anaerobic fermented products. Now, what are the advantages of anaerobic fermentation? production of economically valuable product like carbon dioxide and hydrogen gas can be produced by this anaerobic fermentation process. So it will be it will give more profit to the manufacturer, but there are some disadvantages because maintaining anaerobic condition is very difficult. So we have to spend more money to providing extra provision to the fermenters to like exhaust pump in order to enforce the anaerobic condition. In order to produce vacuum, we have to add some extra provision or extra equipments for the fermenters. So that is the anaerobic fermentation methods. Now coming to aerobic fermentation, as the name indicates, usually this is the normal fermentation process which is usually carried out in the presence of oxygen. Most of the commercial process and majority of the product of human utility is produced by this method. So you don't have to explain more. Normally നമ്മൾ ചെയ്യുന്ന എല്ലാ fermentation processഉം aerobic fermentation method ആണ്. അതിൽ കൂടുതൽ പറയാനൊന്നുമില്ല. Anaerobic fermentation ആവുന്ന സമയത്ത് മാത്രമാണ് we have to remove the oxygen. Okay. Now based on the fermentation occur on the surface or inside the solid or etc. So basically based on the physical state of the culture media, fermentation process can be classified into surface fermentation, submerged fermentation and solid substrate fermentation. Okay, so first one is surface fermentation. So where will be the surface fermentation occur? This will occur only if the substratum is solid or semi-solid liquids. ആ രീതിയിലുള്ള സാധനങ്ങൾക്ക് മാത്രമേ യൂഷ്വലി നടക്കൂ. If the culture media is solid, the microorganism will grow on the substratum and draw the nutrient from the substratum. Solid ആയിട്ടുള്ള സാധനങ്ങളാണെങ്കിൽ ആ സോളിഡ് ആയിട്ടുള്ള സാധനത്തിൻ്റെ മുകളിലാണ് മൈക്രോ ഓർഗാനിസം വന്ന് ഫെർമെൻറ്റേഷൻ നടത്തുക, അല്ലെങ്കിൽ അവിടെയാണ് അത് ലീവ് ചെയ്യുക. ഓക്കെ, സോ basically the desired product are produced by sporulation process. There are some disadvantages for the surface fermentation. Expose the organism to unequal condition like both oxygen and nutrients, because in case of solid nutrient media, the concentration of the nutrient will be varied on the different surface of the culture media. If it is solid, if it is some kind of liquid, it will be uniformly mixed. So the microorganisms are unequal condition for both oxygen as well as nutrient. Now coming to the second one, that is submerged fermentation. The microorganism is grown in the substratum like liquid state. So the microorganism will go into the culture media and it will grow inside the substratum. That is the submerged fermentation. It will be inside the liquid media. In case of surface fermentation, it will be on the surface of the solid media, and in case of submerged fermentation, microorganism will grow inside the substratum like liquid substratum. So the culture conditions are made uniform with the help of sparger and impeller blade. So we will explain what is this sparger and impeller blade. For briefly I can explain. Sparger are the facility which is used to give the aeration, and impeller are the equipments or parts of the fermenter which is used to mix the things. Okay, so we will discuss in detail later. So the submerged fermentation, we will use an liquid state culture media like nutrient broth or sometimes natural products, natural materials in liquid form used as a starting material. So we will make it uniform by using sparger as well as impeller. Most of the industrial fermentation are as of this type. So നമ്മൾ നോർമലി ചെയ്യുന്ന എല്ലാ ഫെർമെൻറ്റേഷൻ പ്രോസസ്സുകളും സബ്മർജ്ഡ് ഫെർമെൻറ്റേഷൻ പ്രോസസ്സായിട്ടാണ് ചെയ്യുക. നെക്സ്റ്റ് വൺ ഈസ് സോളിഡ് സബ്സ്ട്രേറ്റ് ഓർ സ്റ്റേറ്റ് ഫെർമെൻറ്റേഷൻ, SSF എന്ന് പറയുന്ന സാധനം. ഓക്കെ, സോളിഡ് സ്റ്റേറ്റ് ഫെർമെൻറ്റേഷൻ അല്ലെങ്കിൽ സോളിഡ് സബ്സ്ട്രേറ്റ് ഫെർമെൻറ്റേഷൻ. ഓക്കെ, so it is defined as the growth of microorganism and moist solid state in the absence or near absence of the free water. So what is the difference between surface fermentation? Even though the nutrient substratum is solid, that is in the liquid nature, or we will add some kind of solvent into the surface fermentation. But in case of solid substrate fermentation, we will perform this kind of fermentation nearly absence of free water. We will not add any kind of water into this kind of fermentation process. and show much promising in development of several bioprocess as well as product. Solid substrate fermentation. Substrate will act as an carbon source occurring the absence of or near absence of free substance. And solid state fermentation, it will employ natural substrate as above, or an inert substrate which is used as a solid support. So there is a small difference between solid substrate fermentation and solid state fermentation. In case of solid substrate fermentation, we will use the substratum as a carbon source, and in case of solid state fermentation, which will use as a natural substrate as an inert substance, which is used as a solid support only without providing any kind of carbon sources. We will discuss what is meant by carbon source, nitrogen sources, while we will discuss about the media used for the fermentation process. Okay, so these are the different types of fermentation process. Please note, these are the type of fermentation process, not the fermenter. There is another topic. There are some classification of fermenter. We have discussed only the types of fermentation process, not the fermenter. Please note, these are the fermentation process, not the fermenter. The process are of different types, like these are the 9 different types of fermentation process. Now another type of fermentation, another type is there based on the energy metabolism, like type 1, type 2, type 3 fermentation. So basically, from where the energy is produced, that is the basic criteria for classifying this kind of fermentation process. In case of type 1 fermentation, usually the energy is produced directly from the primary metabolites. So that is the difference. In case of type 1 fermentation, energy is produced from the primary metabolites, and in case of type 2 fermentation, the product is formed from the substrate used for the primary energy metabolism, and the product is produced in the secondary pathway. So there will be an primary metabolic products. So during this, by using the primary metabolites, there will be some secondary pathway to produce the final products. Okay, so there is a series of reaction here, type 2 reaction, and type 3 reaction is a clear distinction between primary metabolites and product as they occur in separate step. Okay, type 3 എന്ന് പറയുന്ന സമയത്ത്, even though there is a 2 steps process, it do not depend each other. അതാണ് ടൈപ്പ് 3 കൊണ്ട് ഉദ്ദേശിക്കുന്നത്. പ്രൈമറി മെറ്റബോളൈറ്റിൽ നിന്ന് ഡയറക്റ്റ് ആയിട്ട് പ്രൊഡക്റ്റ് ഉണ്ടാവുന്ന അല്ലെങ്കിൽ എനർജി ഉണ്ടാവുന്നതാണ് ടൈപ്പ് 1 ഫെർമെൻറ്റേഷൻ എന്ന് പറയുന്നത്. പ്രൈമറി മെറ്റബോളൈറ്റിൽ നിന്നാണ് പ്രൊഡക്റ്റ് ഉണ്ടാവുന്നത്. ഓക്കെ, ഡയറക്ട്ലി ഫ്രം ദ പ്രൈമറി മെറ്റബോളിസം. ഇൻ കേസ് ഓഫ് ടൈപ്പ് 2 ഫെർമെൻറ്റേഷൻ, പ്രൈമറി മെറ്റബോളിസം നടന്ന് ഒരു പ്രൊഡക്റ്റ് ഉണ്ടാവണം. ആ പ്രൊഡക്റ്റിൽ നിന്നാണ് സെക്കൻഡറി മെറ്റബോളൈറ്റ് ഉണ്ടാവുന്നത്. ഓക്കെ, സെക്കൻഡറി പാത്വേ യൂസ് ചെയ്തിട്ട് സെക്കൻഡറി പ്രൊഡക്റ്റ് ഉണ്ടാവുന്നു. ഇനി ടൈപ്പ് 3 വരുന്ന സമയത്ത്, ഈവൻ ദോ ദർ ഇസ് എ 2 സ്റ്റെപ്പ്, ബട്ട് ഇറ്റ് ഈസ് നോട്ട് ഫ്രം ദ പ്രൈമറി മെറ്റബോളൈറ്റ്സ്. ദർ ഇസ് 2 സെപ്പറേറ്റ് ടൈംസിലാണ് പ്രൊഡക്റ്റ് ഉണ്ടാവുന്നത്, സെപ്പറേറ്റ് പ്രോസസ്സിലാണ് നമുക്ക് ഈ പ്രോസസ്സ് ഉണ്ടാവുന്നത്. ഓക്കെ, so this is all about the fermentation process. We had discussed about the different types of fermentation. We have defined the fermentation, we have defined the bioprocess, we have defined the fermenter, we have defined industrial products of fermentation, and we have defined we discussed different types of fermentation process. So these are the some of the questions from this area we have already discussed during the initial state of this one. So classify different fermentation method as a 2-mark question. Then operations of aerobic fermentation process. Remaining things will come in the next topics. What is the fermenter, what is the structure of fermenter, what is the structure of continuous fermenter, that we will discuss in the next topic. We will discuss in detail about the fermenter as a next topic. So that's all about the fermentation process. Hope it is clear. Thank you for your patient listening.